Good evening, everybody. I hope I'm audible to all. Okay, good evening. So let's initiate today's session where in today's technique we are going to talk about another technology for DNA recognition for more specific recognition. Okay, so in last class we discussed about the zinc finger nuclease. I hope everybody remember the zinc finger nuclease. Okay, so in the zinc finger nuclease, we have these motifs which are the finger-like motifs associated with the zinc, uh, coupled with the zinc activity and fused with the nuclease. So this particular zinc finger, which is a finger-like motif, which has specificity for 3 nucleotide DNA base. Okay, so each finger could recognize a 3 nucleotide specific sequence. So 2 or 3 zinc finger altogether can recognize a stretch of DNA sequence where this particular protein will bind. And after the recognition, this protein is fused with one endonuclease, which is a sequence non-specific nuclease, but when fused with this zinc finger motif, it with the help of zinc finger, it recognizes that sequence and the nucleases makes a cut at that particular DNA base. Okay, and that is how this entire zinc finger nuclease is evolved and hence being used for the gene editing tool. Okay, so if you have DNA base, a double stranded DNA, you can make a zinc finger nuclease motif where this particular side which recognizes and the other side which is the nuclease will make a cut at this particular position which will break the DNA. Similarly, if you want to make a double stranded cut, you need to provide two such uh, zinc finger nucleases which will make a cut here. This is how the double strand cut will be generated with the help of zinc finger nucleases. And then once the cut is made, you can incorporate any sequence, desired sequence that you want based on homology, or also you can use the non-homologous end joining. So this is how the editing can happen using the zinc finger nuclease. Now today we are going to talk about another such technique where the similar nuclease activity you can incorporate, but the specificity of recognition to the DNA base will be more. You can, so that particular technology is TEL. Okay, so this particular technology, which is TEL, which is transcription activator like effector. Okay, used with nuclease, then it becomes TALEN. Otherwise, generally, we can call it trail protein. So this tail protein is usually derived from a certain bacteria, where these bacteria infect those plants, and inside the plants they induces work as a transcription activator. So when these protein goes and they have a specific recognition sequence, then they bind to that sequence and work as a transcription activator for certain gene, and that's how they induce the transcription of certain desired genes. So these are the how these proteins are present in the nature. So this particular recognition phenomena has been adapted and used and exploited for for more specific DNA recognition. So this talon protein is the alpha helical hairpin domain protein. Okay, which is under tail proteins. So this binds to an individual DNA base. So overall, a single tail protein recognizes a very specific long DNA sequence. Okay, and this is the particular diagram. You can see the DNA double helix, and here the alpha helical hairpin kind of domain of the tail proteins that recognizes a DNA base. So if we'll see the detail of the structure here. So this particular protein, you can see the tail protein are generally, so this is the N terminal of the protein and this is the C terminal of the protein. Okay, so here in between, this is the N terminal region, C terminal region, and then you can see here these dark red color, these are the tandem repeats of the same stretch of some 34 to 35 amino acid stretch. Okay, you can see like from 1, it has been shown here that one repeat has this particular 34 amino acid long stretch. So these amino acid stretch are randomly repeated in this repeat region. Okay, this repeat region is present, and in between the 34 amino acid, the amino acid number 12 and 13 particularly are involved for the specific recognition. So if suppose uh, this particular region here, the 12 and 13 amino acid are N and G, so they can, this particular repeat region, it makes a hairpin alpha hairpin loop, okay, that will recognize only one particular base. Okay, so for example here it is given Ng, its specificity towards the thymidine. Okay, and these is referred as RBT. So the specificity of the repeat is determined by amino acid 12 and 13 position, which are hypervariable residues and are called as repeat variable diresidues. So these 12 and 13 residues are the repeat variable diresidues, and these repeat types have specificity for more than one or more than one DNA base pair. Okay, so if at your RVD position you have these two amino acid, it will specify for thymidine. If you have Ni instead of Ng, it will recognize A, adenine. If you have Nn, the recognition will be changed to Ga. If you have Hd, it will be C. If you have Ns, it will be A, G, C, T, anything. Okay, so this is how those two amino acid are very specific, highly variable, and that provides the specificity for DNA recognition. So there are several repeats, and in those repeats also one repeat recognizing only one. So here Ng is present, that means at this particular position the thymidine residue will be recognized here, the corresponding. If this one is another one and this has this Ni, then it will recognize A, adenine. So that is how this particular recognition takes place. So this RVD base pair recognition follows a simple code, and the individual tail repeats can be genetically linked together to recognize a series of contiguous DNA nucleotide as a single tail protein. Okay, so thus it is possible to use this as a platform for engineering customizable DNA binding proteins. Okay, so the amino acid code for recognition is seen in this particular table. Okay, so if you want A to be recognized, you have to incorporate Ni. If you want C to be recognized, you have to incorporate HT. If you want G to be recognized, you have to incorporate either NN or NK. Yes. NN and NK. If you want T to be recognized, you have to incorporate this NG. Okay. So this is a very useful technique because in the zinc finger nuclease, those are the three uh, nucleotides or three base pair uh, recognition sequence were present, which have many off target sites as well. And here the manipulation of or the recognition you can customize according to your need. So if in your DNA sequence, if you want to regulate the activity or modify or edit one particular gene, and in that gene you know the exact sequence, you can pair, arrange those repeats, join them together in a order such as your DNA sequence. Okay, and that is how you can make tandem repeats accordingly or specifically for your particular DNA of interest. Okay, so yeah. Okay. So this particular uh, so this particular approach or the proteins which are referred as tail proteins, okay, so these are fused with a nucleus. So in the previous session, you remember we discussed about this fork1 nucleus, fork1 nucleus, okay, which was fused with a recognition domain. So this recognition domain recognizes the sequence and fork1 makes a cut there. So these two domains are detachable from each other. So from here, fork1 has been taken and tail proteins have been taken, both together combined. Okay, and that is how the DNA sequence is being recognized. Tail will recognize and fork will create a cut here. Okay, so this is how you can make particular gene editing at the desired locus. Okay, you can fuse it with a nucleus. If not, you can fuse this tail proteins with other kind of proteins as well. For example, you can fuse it with an activator gene or a transcription activator or a repressor. So for example, here in this schematic presentation showed how you can make an inducible system using this particular tail protein. So this tail is fused to a DMRA1. This particular tail protein fused to a DMRA1, and this tail domain can freely bind to the target DNA. So when you want to induce the expression, that time if you'll provide the rapamycin, it will, so this particular VP16, it works as a transcription regulator. So this gene which you want to regulate the expression of, this VP16 can regulate its expression. So upon rapamycin, this VP16 which is fused with this protein DMRC1 and this recognition tail protein which is fused with DMRA1, these both can uh, uh, make a heterodimer bind together, and then this VP16 regulates the activity of this plug protein. Okay, so this was the chemically inducible system. So this is also another chemical inducible tail, but reversibly locked approach. Okay, so reversibly locked approach, you will have the similar thing. So this tail along with VP16 is fused, where at the both the ends you have DMRD. Okay, DMRD. So when given this but which generally interact with each other. Okay, so when they interact, it will be in a close circular form, cyclized form. That time it cannot interact or make the transcription of the particular gene. If you provide rapamycin will bind to these proteins or the monomers and inhibit the interaction between these two. When in an open conformation, the tail protein will bind to the DNA of interest and activate or regulate the expression of this particular gene which is downstream present. Okay. Similarly, here one more TEV sequence is present which can be broken with the TEV peptide, and when broken at this position, then also it makes a linear conformation, and that can also bind to the DNA and regulate the expression. So this is how you can. Okay, so just now we saw that we are fusing this tail protein with several domains, either with repressor, either with a activator, or with the nuclease. So for, and also at the same time, for the specificity of the DNA recognition, we are fusing several repeats of the tail as well, tail protein as well. So this fusion, how can be achieved? So there are several technologies reported, or several methodology has been developed. One of them is a okay, the so that is simple tail assembly reaction star. Okay, simple tail assembly reaction, where you can join several fragments, like two fragments, more than two, eight fragments with each other with very simply. So it is an isothermal approach where you have one particular approach which is referred as Gibson assembly. Gibson assembly approach. Okay. So you have this Gibson assembly approach. So in the Gibson assembly, what you do, you have this fragment and this fragment. These two fragments you have to join together. So how you can join? What you can do in one of the fragment, you can, in this fragment, with the help of PCR method, you can add the certain portion which is overlapping with the sequence where, or the fragment where you want to attach this another sequence. Okay, and once that overlapping sequence you generate with the PCR, what you can do here is, so this is a in a one tube reaction. You, in the Gibson method, what you have three enzymes. One enzyme is an exonuclease, another enzyme is polymerase, and third enzyme is a ligase. So these three enzymes mainly function here. So here the first one is the exonuclease one. Okay. Okay. So another sequence. Okay. So here comes the first enzyme, which is the exonuclease, which function here, which will cleave at the 5' end. Okay. So it will cleave this sequence at the 5' end and produce ends like this. So as there is a similarity between the sequences because there was an overlapping sequence, then what happens? They anneal with each other. The sequence anneal with each other. So these sequences align with each other. Okay. And the gap between these sequences will be filled with the polymerase. The gap will be filled with the help of polymerase, the second enzyme. Okay. So once that gap is filled with the help of the third enzyme, that is a ligase, so the ligase makes the phosphodiester bonds and complete the or locks the DNA. So here the both the fragments that you started with are now joined together with each other. So this is how the Gibson assembly reaction works. It is an isothermal reaction. You can have it in one single tube where three enzymes are involved, and in one go you just need to make an overlapping region between the two fragments with the help of the PCR method. Okay, so let's initiate the session. The question answers. So what is the basic building block of tail protein? So there are four options: beta sheets, helical hairpin, alpha helix, or beta loops. So we just now discussed the structure. Which one would be the correct option? B. Sorry? B. B, helical hairpin. Yes, the structure is in helical hairpin form. Okay, so which of the following is not a physical method of delivery of talent? Okay, so for delivery of these talent or the nucleases, there are certain methods, not certain, like most of the methods of the delivery can be involved, in which the physical methods are there. So there is one method which is electroporation. Electroporation means you will provide a short electric pulse. So you'll put the talent and you will provide the cells where you want to uh, inject the talent. So with the when the electric pulse, these the cell pores will open for few seconds, and with the help of that open pore the talent protein enters. So that is one of the physical delivery of the talent, electroporation. And similarly you have the micro injection. The very small micro injection can be injected to the cell. Similarly, you have ballistic transformation is also one of the physical method. But the transfection reagents, that means there are certain reagents, for example, PEG and some other chemicals that are making the pores in the cell. So those are not referred as the physical method of the delivery of the tender. However, the micro injection, electroporation, and ballistic transformation are all comes under the physical method of the transfection. Okay, so which was it? Okay, skip. Okay, which of the following is an advantage of Gibson assembly? Okay, so we just now discussed. So there is a need for the specific restriction site. So in the Gibson assembly, does it require the restriction sites are required? Okay, so okay, so for normal assembly or joining two fragments of the DNA, there is a more traditional method of using the restriction site, and uh, sticky ends of restriction site can be ligated with each other, and that's how with the ligase you can seal the ends of the DNA, and that's how you can join the two fragments. So that is the conventional method that is being used, the use of the restriction site for joining the two DNA fragments. However, in the Gibson assembly method, we don't need any specific restriction site. Okay, so as I mentioned, if you have two DNA fragments, both you can prepare with the help of polymerase chain reaction. Okay, you take one source of the talent or any specific gene or the domain or the protein you want to amplify. Take the source of that, take the genomic DNA, use or give the primers that are specific, just make the flanking sequence of the other fragment of the gene which you want to associate with. Okay, so with the help of that one simple PCR, you can just put those two fragments and allow the Gibson assembly enzymes to work on a certain condition. So that is a those three enzymes of the Gibson assembly work at this 50 degrees Celsius temperature. where the enzymes are the exonuclease, polymerase, and third one is ligase. So these three enzymes are involved. However, there is no requirement of specific restriction site to uh, join the two fragments of the DNA. Okay, so what is the advantage of the Gibson assembly? There is the need of the specific restriction site? No. Joining of the fragments is specific to the sequence. Can combine only one DNA fragment at once in one tube reaction? No. It can combine more. But the best advantage is there is no need for the specific restriction site, and it will be more easier one-step reaction for joining the two or more than two DNA fragments. Okay, STAR uses an isothermal assembly called DASH. This is a labor and cost effective and accessible, rapid and scalable. Okay, so STAR we just now discussed, and the isothermal assembly also. Which one will be the isothermal assembly? The easiest assembly. Yeah, it will be the Gibson assembly. It is the most easiest and most convenient and cost effective method nowadays. Okay, which animal model or system for investigating human disease such as cancer? Zebrafish. That is the most simplest or most used organism used for the study the human disease. Okay, this is a very simple question. TALEN can be deployed in the form of RNA, DNA, or protein using various delivery method into the living cells. Okay, first is the physical method like ballistic transformation, microinjection, electroporation. All the physical methods can be employed. Bacteria-based delivery can also be employed. Viral-based delivery, there's baculovirus, lentivirus. Chemical methods also utilize the transfection reagents, liposomes, or PEG. So all of these methods can be used for the TALEN delivery. So the correct option will be the all of the above. Okay, when the Lauren and her colleagues successfully create gene knockout using the TAL effectors, the year was 2011. Okay, the full form of STAR is, it will be yes, it will be simple tail assembly reaction. Okay, which of the following is necessary for mitoTALEN to perform gene editing in mitochondrial DNA? Okay, so in the TALEN protein, the C terminal, the N terminal, and the C terminal sequences there. In between, you have those repeat region. So there are several methods. You can fuse towards the C terminal. You can fuse the FOC1 endonuclease, several other type of nuclease, or any transcriptional activator. You can fuse any transcriptional repressor. Okay. Similarly, at the same time, so if you are working on a gene that is being produced or have a nuclear encoded DNA, so what you can do at the end terminal of this particular construct, you can add a sequence, a signal sequence. So suppose you want to regulate the transcription of any particular gene which is present in a nucleus. In that case, you will associate the nucleus signal sequence or nuclear localization signal, NLS. Okay, if you want to send it to the nucleus. if you are working in such a system where you want to regulate the expression of a gene which is a mitochondrial encoded gene, then in that scenario you have to have a knowledge of a signal or mitochondrial targeting signal. So if you fuse this construct of the protein, this entire construct, with a signal that is mitochondrial targeting signal, MTS, then what happens when you uh, transfect this particular construct inside the cell? Here you have mitochondria, here you have nucleus. So if this particular protein has a NLS sequence, it will go to the nucleus. If this particular protein has a mitochondrial nucleation sequence, it will go to the mitochondria. And there it will regulate the expression, either activate or repress whatever sequence that you have attached downstream to this particular tail protein. That function will be performed in that locus or in that organelle. So, which of the following is necessary for mitotalent to perform gene editing in mitochondrial DNA? So, a mitochondrial targeting signal is delivered as donor template for HR. That is not the correct answer. So, donor template for HR, we discussed last time, right? When you are making a break, okay? Suppose you are working this particular protein, NSP1. You have made this construct will form a protein, because this is also under certain promoter. That promoter will make this particular protein from this here. Okay, so when the protein will synthesize, it will break here. Okay, after the break is created, what you need to provide a signal, or sorry, a template, a homologous template, where you can add certain mutated version of the gene or some mutation or some other uh, resistance, anything you can knock in. So if these two sequences are same, so they will perform homologous recombination, and after the homologous recombination, the original sequence is now will be manipulated, and the template that you have provided to repair the broken double-stranded DNA, that template will now be incorporated, and it will disrupt the original gene, and this is the original locus, wild type locus, and this is the modified locus. Something else is inserted, some mutated version of the sequence. It will work as a mutation, it will disrupt the ORF, it will function as a knockdown or knockout of the gene, because the ORF is disrupted. So that is how you can edit the sequence whichever way you desire. So this particular template that you are providing here to repair the break when the break is created is the template provided for HR. So here it is written, a mitochondrial targeting signal is delivered as donor template for HR. So if the template, if you put the mitochondrial targeting signal here in the donor template, that is not going to work. Okay, that is not going to work, because your aim would be to provide the nuclease or this talent. This needs to go towards the gene where your gene is located. So if this particular uh, copy or particular gene is located in the mitochondria, then this protein needs to go to the mitochondria first. So the mitochondrial targeting sequence needs to associated with the talent rather than the template that you are providing for the HR. So this option, the mitochondrial targeting signal, is fused to the N terminus of the talent, will be the correct one, because that is what is required or necessary for the mitotalent to perform gene editing in the mitochondrial DNA. The third option is the microinjection of talent machinery into cytoplasm without MTS. That is also wrong. That doesn't make any sense. Okay, so the second option is the correct one here in our case. Yeah, so these are the basically the questions for the talon. It is a simplest technique, just a modified version of the zinc finger nuclease. The talon, the tail nuclease, transcription activator like nuclease. So if you want to ask anything else from this portion, you can ask me. Otherwise, in the next session, where we'll be discussing more about another technique, which is the CRISPR methodology, where we'll have something different from these two technology, which are zinc finger nuclease and the this talent roach. Apart from these two, we're going to discuss the CRISPR that will be different from this and have more specific and simpler approach compared to these two methodologies. Okay. So in the next class, we'll discuss that. For today, we have only this much. If you want to ask something else, you can ask me. Yes. We are using talent protein for Mm-hmm. Yeah, so this, yeah, so in the beginning, I have shown, na? Like here, you can see this BP16. So, as I have mentioned that you can fuse it with an endonuclease. You can fuse the tail protein with an endonuclease. So tail proteins are have just the basic function of recognizing the DNA sequence. The domain or the functional domain or the functional protein that you associate with it, that will do the exact function. Tail will only recognize at a specific sequence. Okay, so there are n number of genes in your particular organism. Out of those n numbered, you have selected one where you want to make a cut, or where you want to regulate that particular proteins or genes function. So for that, you design the tail protein that will go and bind to specifically to that one particular gene. Along with that, if you associate a transcriptional repressor uh, uh, gene or protein, or a transcriptional activator protein, so with the help of that, the transcription of that protein will be regulated as shown here. So this was an, as it was an inducible system. So in the inducible system, what they have done, they have taken two particular protein which usually recognize or bind with each other with the help of this chemical rapamycin. So you provide or you inject the tail associated with this DMRA. You provide the transcriptional activator or repressor, which is this VP16, which is associated with DMRC. When you are providing this rapamycin, these rapamycin is bringing them close or allowing them to bind with each other, and once they are bind, this all became a one functional unit, and this VP16 is the transcriptional activator of this flug gene. Okay, so this talon recognizing or binding to this protein, VP16 is coming closer to it and then activating or regulating the function of this particular gene. Okay, so that is how you are able to regulate in an inducible manner as well as all in a non-inducible manner, if you will just fuse the transcription activator with the tail. Okay. Okay, anything else? No question, then we'll conclude today's session. Okay, thank you all for joining.